success for EGFR inhibitors in lung cancer. Why have these inhibitors not succeeded in glioblastoma? And I just want to raise one possible explanation. Glioblastoma multiforme, I think is very familiar to all of you, is an aggressive neurological cancer and is almost uniformly fatal with a median survival of 10 to 12 months post-diagnosis. And it was the first tumor type chosen for TCGA. Our group, Jeff Lee, a graduate student in my laboratory, together with colleagues from the Sawyers and Mellinghoff and Sellers laboratory, identified and reported in 2006 a set of EGFR mutations, point mutations, predominantly in the carboxyl terminal domain, including A289 mutations, 596 to 598 mutations, and R108K mutations, in addition to the previously known EGFR variant 3 deletion of the extracellular domain shown here. And yet EGFR inhibitors have not shown efficacy in glioblastoma yet. And here is some more data from Jong-Hee Cho, then a postdoctoral fellow in my laboratory, now an investigator at the Samsung Research Institute, Samsung Cancer Research Institute in Seoul. So Jong-Hee did some beautiful work identifying and then studying carboxyl terminal deletion mutants of EGFR. And here shown in blue are insertion and deletion mutants, and in blue font are the lung cancer mutations, and then in green font are mutations from glioblastoma, including mutations and deletions of the C-terminus and the N-terminus. So with all of these mutations, neither the reversible mutations nor the irreversible mutation, I'm sorry, I just misspoke. Neither the reversible inhibitors of EGFR nor the irreversible inhibitors of EGFR have been shown to be effective for glioblastoma treatment, and nor have drugs against the PDGFRA and KIT amplification. We believe that one explanation is genomic heterogeneity. These are FISH data from a manuscript we recently published, led by Josh Francis and Cheng-Zhong Zhang in my laboratory. And what you can see here in these FISH studies is huge numbers of copies of EGFR in some cells, but very few or in others, or even none. And what this is just enormous heterogeneity between cancer cells in the degree of EGFR amplification, but it's even more complicated than that. So Josh developed the experimental protocols, and Cheng-Zhong the computational protocols to take tumor, dissociate them into single nuclei, amplify and sequence the single nuclei, and then analyze for both mutations and chromosomal alterations in these cells. And they found, strikingly, some extraordinary heterogeneity. So for example, this is another type of deletion of EGFR. It's called the variant 2, and it deletes exons 14 and 15. And you can see, actually, that these glioblastomas, some of them had wild-type cells, some had one variant 2 deletion, and others had a different variant 2 deletion with different length. And when they actually studied these tumors further, it turned out that they arose in different lineages. So each circle here represents a single cell. There were a few normal, non-cancerous cells here. And then there was one set of cells that had deletions in chromosomes 6 and 9, shown here in red circles, and one that did not have these deletions, shown in green circles. And the ones in the red circles almost all had, as far as we can tell, a V2 amplification. In contrast, the green circles either had a V2 extended amplification, or a C-terminal truncation with amplification, or a deletion of exon 25 and 26. Many different types of alterations shown here, and arising separately with independent evolution. So one thing that we conclude here is that the heterogeneity of glioblastoma drivers arises during tumor evolution, and this may be important for glioblastoma therapy because it may take multiple different inhibitors of different kinases or different types of inhibitors of EGFR to treat glioblastoma effectively. And this may be understood by single-cell sequencing approaches, and as I say, it may require multiple kinase inhibitors to treat glioblastoma successfully. Next, I'd like to turn to our sequencing studies of the Cancer Genome Atlas study of lung cancer genomes. The Cancer Genome Atlas aims at the complete cancer genome description of over 11,000 cancer cases with description of both clinical features and genomic features, sponsored by the United States National Cancer Institute. I'm one of 17 principal investigators in this study, which involves hundreds of collaborators. This power of the study is based in part on the increasing power of genome sequencing technology, going from where the cost of a genome sequence has gone from well over $1 billion with the first genome sequence completed in 2001 to on the order of $100 million in 2001, and then the costs following the rate of Moore's law, the idea that in semiconductor and computer technology, the power doubles every 18 months, which underlies the technology revolution that we've seen today, and in fact makes it possible for me to be presenting this presentation to you from here in Boston. And then suddenly in 2007, you can see the rate of improvement of DNA sequencing is beginning to vastly exceed Moore's law, maybe one of the fastest technology revolutions we've seen. And in fact, in 2015, we estimate that the cost of sequencing may drop below $1,000 for a genome. Some of the technical approaches to cancer sequencing include whole genome sequencing, whole transcriptome sequencing of all messenger RNAs, whole exome sequencing of all exons and coding genes, and what we call targeted exome plus sequence, the complete sequencing of exons and rearrangement sites from selected cancer-related genes, such as oncogenes and tumor suppressor genes, which is what, for example, we're doing in the Center for Cancer Genome Discovery at Dana Farber, what I think the Asan Center for Cancer Genome Discovery is doing now, and what we are doing in our clinical test at Dana Farber, the OncoPanel test. These methods allow us to identify the major genomic causes of cancer, mutations, copy number changes such as amplifications and deletions, chromosomal rearrangements or translocations, and infections. Cancer genome alterations can be detected through high-throughput sequencing, as shown here, showing paired end Illumina reads with the ability to find point mutations, insertions or deletions, and then losses or gains of sequence by counting the number of reads. I'd now like to talk about some general features of lung cancer genomes. First, lung cancers are genomically deranged with very high mutation frequencies. We see somatic mutation frequency in this dimension in a log 10 scale, tumor types in this dimension, mostly pediatric tumors on this end, and very high mutation rate tumors such as melanoma, lung squamous carcinoma, and adenocarcinoma of the lung shown here. And these rare outliers with lower mutation rates, those lung adenocarcinomas found in non-smokers. Lung cancers are also characterized by common chromosomal arm level alterations. And what you can see here is sorted by chromosomal position in this dimension and individual samples, each represented by a thin vertical line in the horizontal dimension, with red representing chromosomal gain, blue chromosomal loss, and white approximately neutral copy number. And this slide is from Andy Cherniak in our group. And you can see some striking features. So for example, in lung adenocarcinoma, you see gain of 1Q and 5P. And you also see these in squamous cell lung carcinoma, but you also see very striking, excuse me, please, gain of 3Q and loss of 3P, and gain of 4. And so you see these very striking tumor type specific patterns of chromosomal arm gain and loss. We don't yet understand why these are important, how these alterations contribute to cancer, or how we can use knowledge of them to treat cancer. We also see focal chromosomal alterations. For example, loss of 9P, shown here, harboring a CDKN2A in both adenocarcinoma and squamous cell carcinoma. Gain of 14Q, harboring the NKX2-1 lineage oncogene in lung adenocarcinoma. I'd now like to turn to mutational analysis and driver analysis, targets found from systematic sequencing of squamous cell lung carcinomas. So what are some of the emerging opportunities for targeted therapy in lung cancer identified through TCGA analysis? Beginning with squamous cell lung cancer and our publication in Nature in 2012, work led by Peter Hammerman from the Dana Farber Cancer Institute, working together with my group and colleagues from around the world. Where there had only been a small number of targetable alterations before, Dr. Hammerman and colleagues now found that over 60% of squamous cell carcinomas of the lung have a potentially targetable genomic alteration. And I'd like to highlight a couple of those. First of all, EGFR alterations. They include amplifications as well as rare mutations. The L861Q mutation in histologically proven squamous cell lung carcinomas in 2 out of 178 cases, suggesting the need for EGFR mutation diagnosis in this cancer, and a variety of FGFR alterations in FGFR1, 2, and 3, suggesting the potential use of FGFR inhibitors. I'd like to speak to a couple of these. First, we've seen FGFR1 amplifications in about 20%, first identified both from my laboratory and from the laboratory of my former postdoctoral fellow, Roman Thomas. And seen here in this slide, what you can see excuse me, is that an FGFR1 amplified cell line is highly sensitive to inhibition with the tool compound PD173074, but a non-amplified cell line is not sensitive.